I'm Richard Kitney, shown up here on the slide. That's the red arrows just going over, followed by the Battle of Britain Memorial flight. I feel somewhat intimidated by the competition, but anyway, a few more people might come in. I'm the co-director of the EPSEC National Centre for Synthetic Biology and also the UK's National Industrial Translation Centre for Synthetic Biology. And I'm the co-director because the other co-director is sitting up there in about the fifth row, Paul Fremont. So I want to spend the next 35 minutes or so just talking about why we, or I, believe that synthetic biology will change the world. I want to start off with some background, and the background starts with exactly what is synthetic biology. So in May 2009, we published a report on synthetic biology under the auspices of the Royal Academy of Engineering, and in the report, which was the report of a big inquiry that I chaired and Paul was heavily involved in as well, we define synthetic biology as aiming to design and engineer biologically based parts, novel devices and systems, as well as redesigning existing natural biological systems. So that's the basic definition. Why is everybody getting excited about synthetic biology? Because you'll see in a minute that they are actually getting excited about synthetic biology. And it's because it's seen as a method of moving away from oil-based feedstocks to bio-based feedstocks. So if we think about industry in the 20th century, starting off with synthetic chemistry in the middle of the 19th century, then synthetic chemistry in the 19th century was used, those methods, using oil-based feedstocks to feed into industrial processes, products of many of the great industries of the 20th century. Synthetic biology is an alternative path, if you like, in this process, in this what we call workflow, where you can take a much broader base of feedstocks. You can now go through synthetic biology, through industrial processes to produce products. And here's an example of a couple of products. So synthetic rubber, heavy involvement, this is in terms of synthetic chemistry, heavy involvement in the microelectronics industry. So this is why there's a lot of excitement, because it's seen as the development of a parallel path to the existing path for industrial processes. So next question is, why is synthetic biology important? And this is just one slide which is taken from the World Economic Forum in Davos in 2012, where I'm sure you all know that many of the world's leaders in industry attend this meeting every year. And in 2012, synthetic biology, along with metabolic engineering, but primarily synthetic biology, was defined as the second most important emerging technology for the industrialized world. Now, why is this? Well, it's part of the so-called bioeconomy, which is rapidly developing. So this is one of the people that we work with. This is Rob Carlson, who's a West Coast Californian journalist who specializes in looking at various aspects of synthetic biology. And as Rob points out in this quote, in 2012, $350 billion was the revenue derived from the bioeconomy. And as I say, synthetic biology is becoming an important part of the bioeconomy. So why is this important for the UK? Well, actually, if you were able to go in a straight line, probably about 200, 300 yards from here, you will come across James Watt's steam engine in the Science Museum next door. Now, why is this important in relation to synthetic biology? Well, it's important, and I'm going to just quote here first from Peter Drucker, who's recently died, but a very famous, again, West Coast business guru in this case. In his seminal book, Capitalist Society, he said that within 150 years, from 1750 to 1900, capitalism and technology conquered the globe and created a world civilization. Today, knowledge is being applied to knowledge itself to create the knowledge economy. And it's the knowledge economy that we're very interested in, and particularly, I'll come to this in a minute, in relation to the UK. And here it is. So if we think about the UK, relatively small geographical area, fairly minimal natural resources. We've got some North Sea oil, gas, etc. We want to exploit it. But the people are our true natural resource. We have a very strong, actually, engineering and science base in the UK. And so-called knowledge-based industries are ideal for the UK. And so this is the basis, if you like, from an economic point of view of why we are interested, and many others in the UK are interested in synthetic biology. And I haven't got the slide here, but I should say that a few years ago, probably 2 or 3 years ago, the UK government defined what they called the 8 great technologies for the development of the UK economy over the next 30 years. And one of those 8 greats was synthetic biology. So synthetic biology is very important, as seen by the UK government. So now some background to synthetic biology. I want to start off by talking a bit about the background in relation to biology. And I want to start off by talking about April 25th, 1953. Now this is, what I'm talking about here is the molecular biology revolution. And so on that date, Jim Watson and Francis Crick published in Nature the structure of the double helix, the double helix structure of DNA. And in 2003, Bob May, who was then the president of the Royal Society, described this as probably the most important scientific discovery of the 20th century. By the way, this is Rosalind Franklin down here, who produced the image which Jim Watson then was able to interpret immediately as being of a double helix type structure. Actually, they posed for this photograph next door in the Science Museum 6 weeks after the publication of the paper. And this is a kind of iconic diagram of the double helix, actually drawn by Francis Crick's wife, who was an artist. These 2 images here, or the sketch and the image, represent what's happened, or what happened in the intervening, sort of 47 years from the original publication of the paper, because this is actually now an image of DNA, and that's an image from about 2000. But Jim Watson and Francis Crick said in their paper, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for genetic material. So they spotted this straight away. And then in 2001, we see the initial sequencing of the human genome, the whole genome. And really, I think I would say that this is one of the key markers, if you like, in the development of this new field of synthetic biology. Because this meant that it was possible to read DNA in terms of the human genome and then subsequently in terms of bacteria, etc., etc. So 1953 to 2001 represented 2 datum points in what I would describe as the molecular biology revolution. So that's one, if you like, basis of synthetic biology. The other base is engineering and physical science. And I want to bring out 2 key pioneers, if you like, in this field. First was, that is in the engineering base. First is Norbert Wiener, who was professor of mathematics at MIT in the 1940s. He published in 1948 his seminal book, which was about the mathematics of how biological systems work. It's actually my copy. It's an original edition, which I managed to pick up in Chelsea, in Kensington Church Street for 50p in a second-hand bookshop. didn't seem to think it was worth anything, so I said, I'll pay 50p for it. It's priceless as far as I'm concerned. This was published in 1948. And then the second person in this revolution, if you like, is, as you can see here, Claude Shannon, who in 1948 again published 3 seminal papers on the mathematical theory of communication, actually in the Bell Telephone Lab's technical journal. And if you don't know about this person, he's the guy who did all the work that goes behind this mobile phone, any mobile phone. He was one of the fathers of digital communication, information theory, etc. So I like to think of the second half of the 20th century as being the digital age. So that's not to say it isn't continuing. And 2 of the fathers of that digital age, therefore, were Norbert Wiener and Claude Shannon. Actually, at Imperial College, in relation to information theory and digital communication, Colin Cherry was another major person in this area, internationally recognized. I think that the 21st century will be seen as being the biological age, as typified here in terms of my slide by the original photograph of Jim Watson and Francis Crick, and then the initial sequencing of the human genome. So that's some background. Now, what exactly is synthetic biology? Because I'm aware this is a science festival and we're talking to people from a wide range of backgrounds. So this is one of Paul's slides, actually, and this just shows the so-called DNA sequence and the structure of DNA with the backbones. There is something called the central dogma of molecular biology, which says you start off with DNA, you then strip that out. That's a process called transcription. You produce something called messenger DNA, messenger RNA, and then that translates into proteins. And it's the proteins that build things like skin cells, any other sort of tissues in the body, etc. So this is, if you like, what's called the central dogma of molecular biology. Now, in relation to synthetic biology, why is all this important? Well, these slides, these are again from Rob Carlson, and they just show how the cost of sequencing DNA, so the blue line here is sequencing DNA, how that's come right down in price and actually has gone up enormously in terms of accuracy. So you can see that sequencing DNA is really very inexpensive these days, but it's also accurate. If we look at this yellow line here, there's a dotted section over here. This just shows the cost of writing DNA. So actually being able to write DNA chemically. And we believe that this is going to come down rapidly as well. And then at the bottom of the slide here, we have a number of companies. So GeneArt in Germany, DNA 2.0 in California, etc., who are in business to actually write DNA. And this is now not only possible, but done on a very regular basis. So how does this work in relation to synthetic biology? Well, here are some short sequences of DNA. And what we do in synthetic biology is to define these short sequences because we can actually write them in the way that we want to write them. And then we put them together, and putting several sections together becomes what we call a biopart. You'll hear this, we talk about this a few times. And then typically, although not entirely, we put this into what's called a plasmid. So this new section of DNA goes into a plasmid. And then we introduce this into a cell. We do lots of work on bacteria in this field. So here's an example of E. coli bacterium. And so the plasmid goes in here. And you can think of this as an instruction set. So the DNA is an instruction set which instructs the cell to produce things. So instead of the cell producing what it might naturally produce, it actually produces what we want it to produce because we have we've actually defined the DNA sequence, knowing that that particular DNA sequence is going to cause the cell to do something that we want it to do, or produce something that we want it to do, rather than its natural course of reproduction. So we think of synthetic biology in terms of basic components, these so-called bioparts, and this is what we would call a gene circuit, so it's a simple gene circuit, which comprises a number of different parts. So this is, if you like, the biological description of this. So here's the promoter, ribosome binding site, protein copying sequence over here, and then the terminator. And so that, if you like, is an example of putting a few of these bioparts together. We also think of this in synthetic biology in terms of a hierarchy. So here at the bottom of the hierarchy is the DNA. These sections of DNA are then the parts or the bioparts. They are put together to produce devices. Devices are put together to create systems, and this particular example here is a so-called ring oscillator. So there is this hierarchy that we see in terms of synthetic biology. So here's the biopart. Okay, we then put together a number of these. I've just represented this as 1 to n. So say that is then the instruction set that tells the cell to do something that we want it to do rather than what it would do naturally. And I certainly believe this whole ability to write DNA as well as to sequence it is opening up a major new world, and I really don't think it's overstating it. So this is the Gutenberg Bible from 1455, where there was the move by the creation of the printing press away from writing to actually printing. I think that's what's going to happen in terms of the development of the bioeconomy, because not only can we read DNA now, but we can accurately write it, and that's becoming faster and faster and more accurate and more inexpensive, almost day by day. So we think when we're designing devices of a hierarchy here, and we start off with the parts. These are the components. These encode the biological functions, synthetically designed DNA. These are put together, as I've already said, to make devices. So they're made from a collection of the parts that encode human-defined functions. So biosensors, logic gates, etc. These are some examples of work that we undertake. And then the devices are then put together to create systems which can perform tasks such as counting, control functions, etc. Now, basically, this area, I'm talking about the international field, is essentially work in progress. The parts and devices are now beginning to be well established in terms of lots of them being developed. The systems is the next stage on. So the next question that people often ask us is, why is synthetic biology different? And what's new about synthetic biology? So I'm just going to talk about that for a minute. So synthetic biology provides a framework for the systematic design of biological devices, systems, and in fact in cells, although I'm not going to talk about cells today. It revolves around the application of the engineering principles. So you have to think about this as being essentially an engineering field. It's engineering biology of modularity, standardization, and characterization. And it's the integration of molecular biology, engineering, and computational science, driven by what I've been talking about, this DNA fast sequencing and synthesis. So engineering biology, that's a term which is now being more and more widely used, both here but also particularly in the United States. I want to start off by talking about malaria. I'm not going to go into this in great detail, but this is one of the classic examples of an application area for synthetic biology. I'm coming at it from a slightly different angle. So for a number of years, Jay Kiesling, again, is somebody we know well, who works in Berkeley, worked on the development of something called artemisinin, this drug artemisinin, which is an anti-malarial drug. This is the annual wormwood plant, and it's been known for 2,000 years that this has an active ingredient to be able to treat malaria. What Jay and his colleagues did was to actually modify metabolic pathways so that you could produce artemisinin, or the precursor artemisinic acid, from simple sugar. Now, As I say, I'm not going into this in detail from the science point of view. I want to bring out a separate point, which is if you talk to Jay, he will say it took them 120 man-years of effort, actually by highly skilled metabolic engineers, to achieve artemisinin. Just want you to keep that in mind. Keep that in mind. Let's go back to James Watt's steam engine next door. So the basis of the Industrial Revolution in the UK in the 18th and 19th century was the introduction of systematic processes. That's the key difference between before and after the Industrial Revolution. And this is an important aim of synthetic biology. So I'm going to be talking a bit later on about industrial translation. We're very interested in industrial translation. As I said earlier, we have the National Centre for Industrial Translation here at Imperial College. So this whole process of systematic processes and its importance in synthetic biology is a key factor in all of this. I want to just bring this out in relation to an example. So, I mean, normally we're talking to students, and typically if I'm in a course talking about synthetic biology, I put this up and I say, 35 miles down the road here, down the M4, if you turn off the M4, go right off the M4, you get to High Wycombe, and there in the 18th century, Thomas Chippendale produced exquisite furniture using very highly skilled craftsmen to do that. And what has happened is we've gone from this to this, where the whole process has become systematic, and it's not that skilled craftsmen or skilled engineers, skilled operators have been eliminated, but this is the whole systematic process, and that's what we're working on as an international field in relation to synthetic biology and its industrialization. And we do this by systematic design, modularization, standardization, and characterization. And we build into this the ability to control complexity in terms of biology, but also responsible innovation, so-called responsible research innovation, which looks at the environmental, ethical, and societal issues associated with the designs. Our approach here at Imperial is to develop so-called platform technology, which works across a range of application areas. So here we have the areas that we and others work on, going from biomining through biosensors to biomaterials, etc. And the same basic technology works across that range of fields. And we do this typically by applying what we call the synthetic biology design cycle, where we take into account the responsible innovation issues right at the beginning. We go through specifications, through design, modeling, implementation, testing and validation, and then debugging. We go around this cycle several times before we get the design right. And this is called design, build, test, and learn. That's the paradigm that people use. So a bit about the platform technology. Key areas here are assembling sections of DNA, describing the parts themselves, this is what's called characterization, and defining the cells or the chassis into which they go. So it's this combination of the modified DNA put into the cell, which is key here, and then all of that feeds into an information system which is called SynBio, which is something we've developed over the last few years, and that's just shown there. So parts come into that system, they're stored in a database, and then you can access them basically from anywhere in the world because this is a web-based information system. So one of the key things here is to be able to define these parts as accurately as possible, and that's a process called characterization, and that's something that we focus on a lot. So characterization, as I say, the process of accurately defining how the biopart and then how the biopart and chassis interact. Okay, and I'm just going to give an example of one of the types of bioparts, which is called a promoter. And we typically do this in the laboratory by using laboratory robots. We get data about single cells, we get data about populations of cells, and then we compare these two sets of data. And we typically, for a single part, run this over maybe 3 days, typical example would be 9 runs over 3 days to see what the data actually looks like. And at the end of this process, we hopefully have a pretty accurate description of how the DNA works. This is the characterization process. So this is an example. This is probably my most technical slide. This is an example of looking at what we call constitutive promoters and defining their performance, and we're defining their performance in terms of 2 measurements which are called, or 2 modalities of measurement, so plate readers and flow cytometry, and in this particular case, we're getting some very nice results, and all that information then gets put into the database. And once it's in the database, we can then access it. Anybody in the field can access the information through our web-based information system, which is shown here. So these are so-called data sheets. Typically for a part, there may be 15 or 20 pages of these data sheets, which allow us and other people in the field to actually get lots of information about those parts and the experimental protocols which we use to define the parts, etc. And so these are just 2 sheets. You can drill down into all of this to look at the experimental conditions under the metadata, all that sort of thing. Okay, some examples of applications. So I'm going to talk briefly about logic gates. So if you've got a laptop, anybody's got a laptop, so this is the so-called motherboard, and the basis of this, of any computer, is logic gates. And one of the things we've been working on is the development of biological logic gates, and here's an example. So one of the areas here that we're looking at in terms of the logic gates are so-called AND gates. So we did work with one of our colleagues here in biology called Martin Buck, where we looked at a basic system, which is the so-called hypersensitive response and pathogenicity system. It's basically leaf mold on leaves of plants. This had two channels in its original form. It has 2 channels. We thought if we split these channels and stimulate the 2 channels independently, then we have the basis of one of these logic gates, and that's what we did. And so we actually were able to produce the basis of what we call biological AND gates. And so this just shows the way that AND gates work. So you have 2 inputs, and you have, so these are binary values. You have a 0 and 0 produce a 0, and it's only when you get a 1 and 1 at the input that you get a 1 at the output. So this was we were able to develop this through to a stable AND gate, which looks actually like this. And we did this by balancing the various components in the AND gate. So this one didn't have the right components, so it was asymmetrical, it wasn't working properly. And then when we modified one of these sections here, we got the right kind of response. So this is now an example of a stable device. Why is this important? It's important because biologically based AND gates can be used as for counters, the base of counters, calculators, microprocessors, etc., all in the context of biology. We're not trying to reproduce what we can do electronically, but we want to use this in terms of the biological domain. So a vision here in terms of biosensors might be biosensors that permanently reside in the body, for example, in looking at arterial disease and looking at cancer. So one of our students, for example, who's called James Field, did his PhD on looking at so-called protein nanocages. And if we think about this conceptually in relation to arterial disease, so here's an artery, here's some arterial disease, arterial plaque. The idea here is that we can develop a biosensor which could then be coupled to a drug delivery system because these nanocages are hollow. You can control whether they open or close, and you can put what I call a plaque-busting drug in here. And so this is the basic design. This just shows these biosensors sitting around the arterial plaque. Another example of this, which is now being done in collaboration with the Brompton, actually, is to use a similar approach in terms of cancerous cells. And this is being done in relation to liver cancer. And the idea here is that you can actually cause fluorescence here, and you can actually look at which cells are malignant and which cells are non-malignant. And the idea here is that we can use this logic to build up a biosensor which has, in this case, this is a conceptual idea, but 4 different inputs. Why are we interested in that? Well, it turns out that a lot of these malignant cells have different proteins sitting on their surface, and so if you can differentiate them by biological logic gates built into biosensors, then you can get the appropriate response in terms of a biosensor and then potentially therapy. So now, let's look at some industrial examples. So why are we doing all this from an industrial point of view? Well, the enabling technologies of the DNA sequencing and the synthesis, going through to the core technologies, the bioparts I've talked about, the devices and the systems, leading through to end products in relation, for example, to diagnostics, chemicals, energy, biopharmaceuticals, etc. So if we just think about carbon chemistry, so-called C5 and C6 plant conversion, the idea here is that you take sugar cane. So this is what you see on the television. You take sugar cane, you crush it, you then put it through a fermentation process, and then you can produce bioethanol. That's what happens in Brazil on a fairly regular basis now on a large scale. The problem is that the price of ethanol per gallon is high, and the process uses a lot of industrial or agricultural land. So the idea here is, can we use synthetic biology to improve this situation? So this is the slide I showed at the beginning. Here is the synthetic biology process, the bio-based feedstocks going through industrial processes to products. So this involves so-called conversion processes. So you can start off, for example, with shale gas, or you can start off with general biomass like landfill, as shown over here. You can start off with methane. All of this feeds into a process which produces what is called syngas, and then through syngas, you can go through synthetic chemistry to produce various kinds of products. So this looks exciting because, for example, you can use as the input here landfill. A company called LanzaTech that one of our colleagues is working a lot with is looking at chemicals from captured carbon. So here's industrial waste gases or exhaust gases going through, producing carbon dioxide, carbon monoxide, wasted carbon, wasted energy, etc. And so using the kind of techniques that I've been talking about, you can take this and you can use a bacterial approach using biology and other technologies to produce appropriate chemicals and other industrial outputs. And this looks exciting. And this is done on a cellular basis, so I'm talking now about the LanzaTech process. So here's the cell they use for this process. As you can see, it's a bacterium, and they go through the process as the input, carbon monoxide, CO2, hydrogen. They go through the gas reception process, and then they use a fermentation process based on synthetic biology now, and that's explained down here. Okay, now what about the strategy for the UK? Well, I mentioned the Royal Academy of Engineering report in 2009. Subsequently, we were very involved in a UK government report called the Synthetic Biology Roadmap, which was published in July 2012. And it's this roadmap that has actually represented the basis for actually quite a lot of UK government investment in this field. Oops, sorry. And one of the outcomes of that process has been the creation of the National Industrial Translation Centre for Synthetic Biology, SimbiCity, which is based here at Imperial College. The CEO of SimbiCity is sitting just over there in the audience, Steve Chambers. I'm sure we can talk to you, the audience, afterwards about this. But the whole point of SimbiCity is to create an effective industrial translation engine for bridging the gap between university-based research and industrial processes to create products and jobs through industry, and to develop a national centre for expertise in technology development and commercialisation for the benefit of the UK economy. And we've been at this now for 18 months or so, and it's actually really beginning to work. The whole strategy here that we developed was to base it on 4 hubs, which are essentially the science and engineering hubs. So this is where all the processes go on, all the projects. A facilities hub, and we're building now what we call a foundry for producing DNA, an applications hub, and then a business hub, because not only do we want to do the science and the engineering here, but we also want to help with the creation of the business expertise in relation to the translation of synthetic biology. We have a range of industrial partners, so these are some of the multinationals. There are about 9 multinationals that we're working with now. And we have of the order of 40 SMEs as well that we're working with. So this is pretty intensive work that goes on a daily basis, all about taking university-based research and translating it into UK industry. And Imperial College is leading a range of academic partners, Cambridge, Edinburgh, King's, Newcastle, and a number of other UK universities in this process. So we act as if you like, the catalyst for the whole of the industrial translation process. And the Industrial Translation Centre is called SimbiCity, and it's got a range of functions, training, funding, project management, etc., to translate university research into industry. But we're also very interested in building the workforce. There's no point in building lots of exciting technology if you don't have the appropriate workforce. Oops, sorry. So within SimbiCity, the industrial translation process, we have an educational pipeline, and this is all about business and outreach. We run a 3-day MBA. We run something called Lean Launchpad that I'll come back to in a moment. We also run with colleagues of ours in the United States something called LEAP, which is a leadership accelerator program for highly talented young people who are moving into the field of synthetic biology. The Lean Launchpad is a really exciting course. We partnered with Steve Blank and Jerry Engel from, so Steve is from Stanford University, Jerry is from UCSF. We've worked with them to develop this course for synthetic biology. We've had a very successful first run at this. This course is about company development, business models for the design of products, etc., and customer development. And it's a very exciting course, which we can tell you more about offline if you're interested. So in terms of the actual education pipeline here at Imperial, because Lean Launchpad is done and the other course is done within the Industrial Translation Centre, but then we have more formal courses going on in undergraduate, postgraduate, and continuing education within what we call the Imperial College Synthetic Biology Hub. So Paul and myself in the lab, so we successfully run an undergraduate module. We've had 200 students going through that. This is a final year module. We also run a master research PhD program in systems and synthetic biology. We've had 60 graduates since we started this in 2020. So this is a more formal educational process. And finally, we typically have an Imperial College team in a major international student competition, which is called iGEM. This is from 2014, where we came second in the world out of 220 teams. We've been in this actually since 2006, and we've never been outside the top 6 teams in the world in terms of this competition. I put this up particularly because if you go over to the main marquee, you'll see a big exhibit where the students are over there talking about 3 of these separate iGEM projects. And I hope you'll do that because they're really enthusiastic young people who've got a great deal of very interesting things to say. Okay, so some concluding remarks here, just to wrap up. First of all, the financial world take this sort of thing very seriously now, and you can see that in relation to, so this is Nature Biotech, from Nature Biotech, actually from February this year, and you can see what's happening in terms of the investments, in biotechnology, which, as I say, synthetic biology feeds directly into. So financial investment in this area is increasing rapidly. This is my final slide. This is somewhat speculative, but it shows how, in terms of the microelectronics industry or the electronics industry, starting off in 1950, there's been this increase over that period from 1950 to 1990 through to 2000 in terms of revenue, in terms of billions of dollars coming from initially the transistor, then large-scale integration, and then very large-scale integration in terms of microchip technology. What a number of sources, economic sources, if you like, think is going to happen in terms of biotech, and as I say, a really important driver of biotech is synthetic biology, is that we are now beginning to see, in terms of revenue, a pretty rapid increase, and you can see synthetic biology right there, which is why we and everybody else is really interested in the whole area of the bioeconomy, developments of the bioeconomy, particularly in relation to the UK economy, but also in relation to this, what we believe, key industrial driver, scientific and industrial driver, called synthetic biology. So thank you very much for listening to me.